Okay, great. So I think that we can get started. Hi there, everyone. We are so happy to have you, whether you are joining us here today or you'll be watching later on the Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already and include any questions in the chat box below. We will have time to answer questions at the end of the presentation. My name is Monica Joanna Alnacave, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with a sole focus to develop potential therapies for GABA-A variants. By partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trials. Today, we welcome Dr. Mu, who recently joined us at our inaugural Cure GABA-A Scientific Roundtable, which was held in conjunction with the American Epilepsy Society. Dr. Mu is an associate professor with a PhD in biochemistry from Case Western Reserve University School of Medicine. He has a rich academic background from the prestigious Caltech. He is currently leading the Mu Lab, centered on neurotransmitter-gated ion channels, with a specific focus on GABA-A receptors. In simple terms, for us non-scientists, the Mu Lab is focused on studying specific components of our nervous system known as neurotransmitter-gated ion channels. These channels, including GABA-A, act like tiny doors in our brain cells, influencing the balance between excitement and calmness in the central nervous system. If these channels don't function properly, it can lead to conditions such as epilepsy, autism, and intellectual disability. The Mu Lab aims to understand these channels better and to find ways to treat or prevent these neurological disorders. I'm handing it off to you, Dr. Mu. Please share with us your presentation on GABA-A receptor variants and the potential therapeutic strategies.
Okay, all right. So first, I really want to thank Monica for giving me this opportunity to present our research about the GABA receptors. The hope of this presentation is to give more information about this GABA receptor clinical variants, how we can use our current knowledge to develop some potential therapeutic strategy for the treatment of the genetic epilepsy resulting from GABA receptor dysfunction. Okay. So this is outline of my talk today. So basically, we will follow the thread from the, let's see, whether we can get this. No, okay, that's okay. We'll follow the thread from the physiology of GABA receptors to the disease-causing mechanism and to the potential development of the treatment options for GABA-A variants disease. Basically, we will give a background about the GABA receptors and their clinical variants. Afterwards, we want to discuss the disease-causing mechanism for this clinical GABA-A variants. After getting to know the principle of this disease-causing mechanism, we want to share our study about how we might be able to develop some novel therapeutic strategies to correct the function of pathogenic GABA receptor variants. We will start from the background about GABA receptors and their clinical variants. So if we have a look about the signal transduction in the human brain, we will get some basic idea about how the brain cells communicate with each other. The brain has a really a huge number of millions and billions of these basic building blocks called the neurons. The neurons basically is a nervous cell. It has the soma has many of these dendrites that are responsible for the receipt of this the signals from the adjacent neurons, and also the neuron have a single axon. It is a really long projection into the neighboring post-synaptic neurons. So the neurons communicate with each other through a combination of electrical as well as chemical signals. When a nerve impulse travels along this very long axon, it reaches its terminal. Then this terminal has these really tiny gaps. These tiny gaps is called a synapse, which can be illustrated in this figure. Then afterwards, the neurotransmitter, which are small molecules that are responsible for the transmission of the chemical signals between two neurons. Neurotransmitters will be released from this presynaptic neuron and diffuse within milliseconds across these synapses and bind to the post-synaptic neuron. This is another the neighboring neurons. Then the binding of this neurotransmitter to their corresponding receptors, for example, the inhibitory GABA receptors or the excitatory glutamate receptors, the binding of this neurotransmitter to this post-synaptic receptor will induce the downstream signaling pathway. In the case of ion channels, this ion channel protein will open, the ion pore will open, then chloride, the different ions will move across the membrane, inducing the downstream signaling pathway. This is the basic idea about how signal transmission occurs in the brain between different neurons. To understand about the basic idea about how system might occur in a patient, so we need to have a basic idea about the so-called excitation inhibition balance in the brain. Basically, for the brain to function well, it needs to maintain a delicate balance between the inhibition behavior and with this excitatory behavior. Seizures can be induced either by the enhancement of this excitatory stimuli, which is mainly regulated by glutamate receptors, or by the impairment of the inhibitory mechanism, mainly governed by GABA receptors. The process of integrating different types of input signal, basically the excitation was inhibition, lies at the heart of the neuron communication. The neuron is a tug of war between inhibition and excitation. So the take-home message is that if you the brain has a tendency to be hyper-excitable, it might has a tendency to produce seizures in the patient population. All right, so now so we for Monica set a really nice foundation, the Cure GABA-A Variants. So let's talk a little bit more about the GABA receptor. This another background about this so-called GABA-aminobutyric acid type A receptors. It is a major inhibitor ion channel protein in the human brain. It's responsible for the regulation of the vigilance, anxiety, epigenetic, and activity and memory function. In human, at least 19 subunits has been discovered. Loss of their function is known to lead to a variety of neurological disease, such as epilepsy and intellectual disability. So for these 19 subunits, people have studied their abundance in different areas of the human brain. Basically, people use iron-seq or iron profiling to determine the right abundance of different subunits. We can see from this across this line is different brain areas. Like, for example, this is the cortex, this is the thalamus, middle brain, cerebellum, and the hindbrain. So basically, we can see that GABA receptor subunits are distributed throughout the brain areas. GABA receptor is everywhere in human brain. But we can also find some of the major subunits of GABA receptors. For example, the alpha-1, beta-2, beta-3, or gamma-2. Basically, this is also the major subtypes of GABA receptor in the human brain, composed by alpha, beta, and gamma. Okay. All right. So if we with the development of genetics sequencing, recently, especially during the last 10 to 13 years, people have identified many of these epilepsy-associated GABA-A variants. For example, if we go to this NIH ClinVar website and type the GABA-A, GABA-A1, basically this is the alpha-1 subunit of GABA receptors. If we type this one, this will pop out about the currently known GABA-A variants in this alpha-1 subunits. Let me see whether I can just get my names. This is okay. From here we can find this whether the variants has some clinical significance. And also we can find that indeed for this alpha-1 subunit, there's over 200 missense mutations have been identified. So probably I can show you, probably this is very nice website. For example, I can give you some of this example about how you might be able to find whether the mutation or the variant of this GABA receptor has identified in the population. We just determined about this GABA-A1. Then if you type GABA-B2, this is beta-2 subunit GABA receptor. Basically, you can find this. This website will be able to give you this really nice information about this comprehensive list of the currently known GABA variants here. For example, if we click missense here, it will give you all the information about what is known for these 219 variants in the GABA-B2 variants. This is give you the information about this location in this protein sequence, and also some of these the conditions that associated with the mutation. And also clinicians or the research have given some like the annotation about whether such mutation is categorized as significant. For example, many of the, you can find that, let's see, many of these mutations are categorized as unknown, uncertain significance. Part of the reason is that many of the mutations have not been studied extensively. Probably only the clinician identify this mutation and subscribe this mutation to this NIH ClinY database. And also you can see some of these mutations are categorized likely benign. It means that most possibly this variation will have limited effect on the function of these GABA receptors. Correspondingly, benign variations might not lead to disease phenotypes. Okay, you can, if we go through this list, we'll find that many of these variants are still have unknown significance, right? Okay. All right, so this is how you will be able to check the currently known information from the NIH ClinY database. It's maintained by the National Institute of Health. So it is really has pretty rich information. So if you want to escape the typing, searching for this different gene, you can our lab also summarize all these different subunits for the GABA-A variants. It can be direct. You can use this direct link here. You will be able to find all these subunits variants in this location. All right. So this slide basically summarize what is known for the major subunits of GABA-A receptor subunits. Remember that the major subunits of GABA receptor are alpha-1, beta-2, beta-3, and gamma-2. We can find that according to the NIH ClinY database and their annotation. For each subunit, for example, for alpha-1, there's over 200 missense variants. Among these identified over 200 missense variants, almost 170 is still needs to be characterized for their clinical significance. It's the same case for the major subunit beta-2, beta-3, or gamma-2. For each major subunit, over 200 variants have been identified, but the majority of these variants still need to be further characterized. According to this number, over 70% of variants still need to be studied or be characterized for their clinical significance. Whether that's variants will lead to functional defects is still unknown for the majority of these clinical variants. So basically, this is a cartoon figure shows the overall organization of the GABA receptor proteins. Basically, the major subtype are composed by 2 alpha-1, 2 beta, and 1 gamma-2 subunits. If you rotate this stretch 90 degrees away from us. This will be another view, side view of this GABA2 5 subunits. Okay. All right. So for each individual GABA receptor subunit, it has this. This is the summarized protein sequence of this cartoon for this primary protein sequence of GABA receptor subunits. It has this N-terminal, really large N-terminal part. It also has 4 transmembrane helices embedded in the lipid bilayer in the cell membrane. We summarize here in this primary protein sequence are these different our current knowledge about whether position contains pathogenic variation or uncertain variation or the benign variation. I know this is a really rich of the information. For example, if we focus on this gamma-2 subunit, if there's variations occurred in the NIH ClinVar, this will be highlighted as red. We can find that for this transmembrane domain 1, 2, and 3, they are rich in this pathogenic variations for these subunits, indicating that this transmembrane domain are really a hotspot to contain the pathogenic variations. And also there's some loops connecting different parts of this GABA receptors. They also have this so-called hotspot to harbor this GABA receptor variations. Okay, but from this look at this sequence, you will find that many of these positions are colored in yellow. Yellow means uncertain clinical significance. There's we need to future research need to be done to characterize this the functional outcome for this variants. But before doing the actual experiments with advance of the recent artificial intelligence, we will be able to predict whether given variations in GABA receptor position is potential pathogenic, potential benign, or not. So basically, current we utilize 2 state-of-the-art AI-based, artificial intelligence-based algorithm to predict every single variations in all the positions of GABA receptor subunits. The result is summarized here. We can see some of the colored dots on this figure, right? So this all this red, yellow, or green colored dots indicates the red variant that have been annotated in the NIH ClinVar. But you can see that on the background, many of the over 4,000 dots are still had not been characterized. Surely, not all these variants has been identified clinically, but it just shows that with AI prediction, every possible variation in the GABA receptor subunits will be able to get some prediction. Okay, all right. So this figure probably is really complicated, but for example, if a patient come to us and asking us what is the potential pathogenic probability of this given mutation, I included as a step-by-step tutorial so you will be able to do it on your own computer to determine whether a given variants in this GABA receptors are pathogenic or not. So all these databases are public available. You will be able to do it at home on your own computer. For example, the first step is to go to this website, because this is the web server host this publication. Then paste that link to any of this internet browser. You will find this is a pathogenicity prediction of GABA receptor variants. From this, you can find that we can click on supplemental materials because this table is really huge. It cannot go to the main text. If you click on this supplemental material here, this will lead to all these four supplement tables. From these four supplemental tables, table S1 is for the alpha-1 subunits, S2 is for beta-2 subunit, S3 is for beta-3 subunits, S4 is for the gamma-2 subunits. For example, if we want to check the variation in the beta-2 subunit, we can click this download. This will download this Excel table to your own computer. In this case, it has name of this median 2. Then we go to the file that was downloaded. Then you can open this Excel file. Okay. After you open this Excel file, it will give you the AI predict pathogenic probability for any of these given variations in this receptor. Okay, all right. So this is pretty data rich. Let me just go through this one with you. What you really want to see first is about the column A. Column A is basically variations that's a patient might would harbor. For example, this A304V variations. Then the next thing you want to focus is this alpha missense variation. This is column C. Column C is a prediction result from this AM-based prediction software. Also you can use column E. This is a prediction result from another AI-based prediction software. Another column of interest is column H, because this column H basically is annotation from the NIH CleanWire database. For example, I can give you an example here. For example, if in column H for this specific row 39, sorry, this is highlight this one. For column H, we can see that for CleanWire annotation, it said that the annotation of this variation is pathogenic. Indeed, for the prediction of the AM, that it is also described as pathogenic. But for this Rhapsody prediction, it somehow is predicted as neutral. There's some difference between two different predictions. But overall, these two predictions are pretty consistent with this current known annotation from the NIH. The really nice feature about prediction is that if you go to some of this, for example, for this N124S variations, currently the NIH, it did not give us any information about this significance, but both of these prediction are stating that this variant is pathogenic or deleterious. So this is we are pretty confident most possibly this variant will be pathogenic. Surely this it needs to be further verified experimentally. For this prediction algorithm, you can, in addition to currently known CleanWire data, it also can predict any of the potential variations which currently has not been identified yet. Therefore, this database will be really nice resources for the community to check any of the potential variations in this GABA subunits. Okay. All right. Okay, now we can go back to this next topic. Next, because we now identify people identify many of these clinical variants. Now we want to determine why this variants might lead to dysfunction of the GABA receptors. That is a disease-causing mechanism. So for now, we can go through this cellular signaling pathway briefly. So for all membrane proteins and the proteins in the secretory pathway, they are synthesized into this cellular organelle called the ribosome. After protein synthesis, membrane proteins they need to be translocated into this cellular organelle called the endoplasmic reticulum of the ER. It is within the ER proteins need to be folded into their proper structures. Only after the proper protein folding steps, this protein will be able to exit into this protein factory, go through another organelle called the Golgi apparatus. Afterwards, their final destination will be reaching the plasma membrane for their function. So in short, membrane proteins they need to be present in the plasma membrane. If they are not present in the membrane, they will not function, right? So it's also known that membrane proteins, if something go wrong with this protein manufacturing steps, they will be retained inside the cell and degraded by another cellular organelle called the proteasome. The final result is that if a protein cannot go to the membrane for their function, they will be they will not have sufficient function. That's two different disease phenotypes. Okay, so basically the protein homeostasis network regulates the protein synthesis, folding, trafficking, and degradation. This is also reflected in a healthy physiology. The disturbance of this protein homeostasis in cells lead to different diseases. Okay, so for ion channel proteins, in addition to GABA receptors, in the literature, people have studied different types of ion channels. For example, the excitatory NMDA receptors, and also people studied another ion channel proteins in the lung called the cystic fibrosis transmembrane conductance regulator. This CFTR is responsible for the disease causing of the cystic fibrosis. People have studied very extensive about why CFTR misfolding can lead to cystic fibrosis, and people discovered many of the correction methodology to treat cystic fibrosis. Okay, so this is recapitulation about this disease-causing mechanism for those GABA receptor variants. Basically, these GABA receptors they need to form this 5 subunit pentamers in the endoplasmic reticulum. Only after this assemble this pentamer, this receptor can traffic to the plasma membrane. If for each individual subunit, then to go through this protein folding step or protein assemble step. If something goes wrong with the protein folding or assemble step, this GABA receptor they will be retained inside the cell. Therefore, they are subjected to excessive degradation. No sufficient protein will be present on the membrane for their function. Okay, then for the over 1,000 clinical variants in GABA receptors, we can categorize these variants to different classes. This is important because this, if we get to know what are the molecular defects for these variants, we will be able to target the underlying molecular mechanism using different strategies that will be more efficient, effective treatment. For class 1 is basically the proteostasis defect. For this class of the variants, these proteins, GABA receptor, they cannot reach the plasma membrane. Remember, plasma membrane is a function of as location for GABA receptor. If that cannot reach the membrane, that cannot be functional, right? So this class 1 means that the protein either have some protein folding defect or protein assemble defect or protein traffic defect. The net outcome is that no sufficient membrane protein will be present on the membrane. The class 2 is that refer to the variants that have the electrophysiological defects. It means that the protein can be present as a membrane, but the protein are functional deficiency. There's some of the GABA receptor contact chloride, right? So there would have some kind of the channel gating defects. No sufficient of the chloride will move across the membrane, even if the protein are present on the membrane. The class 3 is more like the early MI level. So if have this so-called nonsense or frame shift mutation, this will produce a premature termination of these GABA receptors. There will not be sufficient of proteins are synthesized in the ribosome. Therefore, there's no sufficient proteins produced. Also lead to functional defect for these GABA receptors. The class 4 will include all other disease-causing variants with remaining causes. Okay. Then, so as example, we choose to study selective alpha-1 GABA receptor alpha-1 subunit variants. That's over 200 variants, right? So we selected 8 variants according to their special distribution along the primary sequence, either the N-terminal domain, transmembrane domains, and also we select the variants according to their disease pathogen probability, ranging from moderate to severe. We want to cover the variants as broad as possible. For example, this is variants that are studied in different in this transmembrane domain. The variants studied in the N-terminal domain. Okay, so from our study, it's very clear that for all these selected alpha variants, that will cause loss of function. If we compare their activity to the wild-type GABA receptors. So we basically use electrophysiology PET clamping recording to study the function of this GABA receptor. This is a GABA-induced chloride currents. The black curve represents the wild-type GABA receptor. The application of the GABA will be able to reduce very fast and robust current as shown here, but if the variants, in the variant case, we can say some variants will lead to a significant or substantial reduced GABA-induced current. In some cases, have the relative moderate effect. Some variants will cause the reduced current, but kind of in the middle between the really severe variations as the moderate variations. So, and also the chloride current is peak current is one measurement. Another measurement is about the potency for this for the ligand, because the GABA receptor bind GABA. So sometimes the variation also lead to reduced receptor sensitivity to the ligand GABA. So basically, for this is dose response curve for the wild-type receptor. Some variations will shift this dose response curve to the right, indicating that the ligand GABA is not as sensitive in this variant's case compared to the wild-type. Okay, so the take-home message is that for all the select variants, they will lead to functional deficiency. And also we continue to determine why these variants lead to the reduced currents compared to wild type. So basically we want to determine whether these variants will lead to reduce the protein expression level compared to wild type. On the left, we show that indeed from many of the variants that reduce the total protein level for these GABA receptors. On the right, we show that most of these variants will lead to reduced surface expression for this for GABA receptor, indicating that if this mutation of this variant occurs, this GABA receptor have a reduced presence on the plasma membrane, indicating indeed the protein homeostasis defect is one of the major disease-causing mechanisms for these GABA receptors. So this is important because this is also influence how we target our therapeutic strategies. Again, this shows that indeed this is one example of this variant A322D in the alpha-1 subunit. Immunofluorescence staining determines whether this protein GABA can be present on the cell surface. In the wild-type GABA receptor, we can see we can have a really clear surface presence here as represented by this red staining. But in the case of this variation, so basically we cannot find any of the surface staining indicating all these mutations. These variants can degrade very quickly inside the cells. Therefore, this receptor cannot reach the plasma membrane, leading to really severe loss of function for this variation. Okay, now we have some discuss about the disease-causing mechanism. Now we will, according to the disease-causing mechanism, we will be able to target the strategies to treat this functional deficiency using different strategies. Okay, all right. So let me see how we can pull this. All right, now it's much better. All right, so basically, what we discussed is that many of these probably maybe 60%, 70% of the variants have this proteostasis defect. This defect is caused because this due to this inefficient protein folding step. We can think that this DNA transfer into RNA is translating to protein. Protein need to go to their functional state. We can see this is a black box. If something goes wrong with this black box, we will not be able to produce functional channels, right? So the strategy we are taking is that how we can correct this misfolding of this trafficking deficiency for this GABA variants. As this in this case, we will be able to restore the trafficking these variants to the surface. This will represent a novel therapeutic strategy, which is also important because currently many of these drug-resistant GABA variants are due to a current antacids drug is because those antacids drugs only target GABA receptors once they reach the plasma membrane. But if at the first place, the GABA receptor cannot reach the membrane, then those antacids drugs will not be able to function very effectively. So for our lab, we try to use two different strategies. The first strategy is called we use so-called proteostasis regulators. Basically, this kind of small molecule, they are folding enhancers. Those folding enhancers, they will be able to promote this cellular protein folding capacity in general. Because the variant, GABA receptor variants, they are destabilized, it will be able to be differentiated from the wild-type proteome. The enhancer protein folding capacity will be relatively specific to target the destabilized mutant proteins as a way, the application of the proteostasis regulator will be able to refold the otherwise degraded proteins, enabling their trafficking and assemble to the plasma membrane. The network outcome is enhanced functional surface expression on the surface. And also, if the variant can reach the plasma membrane, their function is partially restored. In many cases, it can reach the clinical significant level. Okay. So the second strategy we are using is that so-called the pharmacological chaperone strategy. These pharmacological chaperones, they are GABA receptor specific. This basically is a small molecule that can direct interact with GABA receptors, the interaction of these small molecules will be able to stabilize the variant, therefore enabling their assemble into the functional pentamery receptors and forward to the plasma membrane. This also will be able to restore their functional surface expression level. Both strategies have been have their advantage and to rescue the function of the GABA-A receptor variants. Okay, so for pharmacology chaperone strategy, basically this we use a screen method to screen out the known library of this GABA receptor direct interaction proteins. From this screening, we find several really potent lead compounds that can increase the protein level of the GABA receptor in different cases, like the DN mutation, GD mutation, PL mutation. Among these top compounds, we found that hispidolin and TP003, they are among the most effective compounds to increase the GABA receptor variant protein expression. Surely, the application of these two small molecules will be able to increase the surface expression of these GABA-A receptor variants. Surely, we continue to test how this will influence their function. Indeed, the application of these two small molecules can increase their function using patch climbing recording. Okay, since GABA receptor result in the central nervous system, we continue to develop iPS-derived neurons as a system that most natively mimic the patient environment. So we basically knock in this GABA variant into iPSC, then differentiate this iPSC into neurons. Then we use immunofluorescence imaging technique to determine whether the application of the small molecules can increase their expression on the cell membrane. Okay, so that's first class is a molecule called the pharmacology chaperones. The second class is proteostasis regulators. Indeed, we also identify several classes of proteostasis regulators, include the UPR activators, the calcium channel regulators, and some of the approved drugs such as dinoprost and dihydroergocristine. So basically, to identify such small molecules, we use high-throughput screening to develop this cell-based assay and screen this FDA-approved drug library. From this limited screening of this drug library, we identify the top two lead compounds. One is called the dinoprost. The other one is called dihydroergocristine. So these two compounds can both cross the blood barrier. DNP is prescribed to induce labor now. DHEC is currently used to reduce blood pressure and treat dementia. Okay, and also application of these two small molecules will be able to increase the function of these GABA variants in various cases, but their effect on mutant receptors are pretty specific because they don't influence the wild-type receptor very significantly. This is important because we want the proteostasis regulators be specific for the variants, but without influence the wild-type protein too much. Okay, so in summary, for today, I hope that I show that that the reduced surface trafficking and the loss of function of the GABA receptors is a major disease-causing mechanism for GABA receptor variants. Restoring proteostasis pharmacologically corrects the surface expression and the function of the pathogenic GABA receptors, representing a promising therapeutic strategy to treat GABA receptor variant related to genetic epilepsy. Several lead compounds such as hispidolin and dihydroergocristine were identified as effective reagents to correct the function of the GABA variants. Their further development is needed for a translational application. So finally, I want to thank my lab member. So this basically they as a hero that carry out this really this kind of research to push this research field forward. We are also thankful to our collaborators from different universities like Dr. Kelly, Dr. Weizmann, Dr. Katie Kang, Carol Meadows, and Ran Nashmi, Ashley Sheffer, Chris Richards. They help with the different assays development. And also I want to thank the funding from the NIH to support this research. And I thank you all for your attention, and I would be happy to take any questions.
Thank you, Dr. Mew, for your presentation and for touching on the topic of variants of unknown significance and the introduction of the AI for the use of the prediction tool. Cure GABA-A will include the BioRx link in our website and the instructions. But we did have a question from Teresa, and she was asking if the AI tool also includes nonsense mutations.
For that current alpha missense that are not for the nonsense mutation, for the nonsense variation, that only include the variations for the missense. That's basically that single single amino acid substitution. But hopefully in the near future, the AI field will advance to extend the prediction for nonsense frame shift variations. Right?
Okay, that's great. And I also wanted to mention that we partnered with Combined Brain, and we've been collecting biosamples. And I wanted you to touch on why it's important to have biosamples from a patient's perspective.
I think that's really valuable and probably the really timely step to push this research field forward. Because for our this have probably three aspects that's really valuable. The first is that this will organize the patient community. Then while doing this sample collection, this patient will be especially this foundation will be able to gather this information for the parents, for the patients. It will be ready for future clinical trial. Because then when the clinical trial starts, it will be able to access the patient population. This is really important. The second one is about the research field, because currently many times we only use the cell culture system or the mouse model system. We don't have the samples from the human. Sometimes people say that the human human sample is the most relevant system to study this GABA, this variants, not only for GABA variants, for many of these protein variants, right? So if we can get a sufficient amount of this sample, and this will be really valuable to the research community. For example, we are now making a really huge effort to use iPSC-derived neuron to study GABA variants, because we think this is the most native environment to mimic real patient environment. If we can get more information or get more samples for that purpose, this will definitely makes our research and discovery most relevant to the patient population, right?
I agree with you, and it will move things forward much faster. Our goal in the coming months is to collect as many biosamples as we can through Combined Brain. So we are so excited for that partnership. I also had another question here from Laura. She's asking, are there any specific breakthroughs or findings that we can share with the patients regarding gene mutations? Now, I know you were touching on correcting the folding and trafficking deficiency. Your research includes 2 strategies using 2 different types of small molecules. I'm very excited to see this develop. My question to you would be, are you currently using this or testing it in mouse models?
First, I want to say something about the genetic variants, right? So for genetic variants, I think the first clue is that you will be able to use AI prediction that will give pretty good idea about whether that's potentially pathogenic or relatively benign, right? So then, secondly, is that for the testing, this is small molecules. We haven't gone through the mouse model for that testing yet, right? But we have done using this small molecule in the iPS-derived neurons, right? So I think for in the at least in the cell culture model, I think that will be sufficient, sufficiently to mimic the native patient environment. Because mouse are sometimes different from human, so it's not. I mean, so they correlate, yeah, but sometimes they don't. But it's good. I think it will be good to have both the cell culture data as well as the mouse model data. Yes.
Which brings us back to biocollection and why it's so important. But Dr. Mew, I just wanted to thank you so much for joining us and presenting today. Thank you to all of our caregivers who joined us and to the ones who are joining us on YouTube. We really look forward to everything that you will, all of the science that you will develop in the coming months. And we look forward to talking to you again. Thank you, Dr. Mew.
All right. Thank you.
Bye-bye.
Bye.